We're now going to discuss the synthesis and reactions of nitriles. Nitriles are very readily synthesized by SN2 reaction. The reaction itself looks like this. We start with an SN2 substrate, Rx. We treat it with sodium plus, cyanide minus in DMSO. We use DMSO because it's a polar aprotic solvent which favors SN2, and we need to be able to dissolve that ionic substance. We get an SN2 reaction where the cyanide reacts with R, we push out X minus, and we get this product. Now, because this is an SN2 reaction, we need to use an unhindered carbon group in our substrate, and so therefore this is limited to methyl groups and primaries, and in theory unhindered secondaries, although in practice cyanide is a pretty strong base, and so therefore it's very hard to get unhindered secondaries to go. We normally get mostly elimination. The mechanism then is essentially a classic SN2 mechanism, which we discussed last semester. We have our substrate, the cyanide nucleophile approaches from the back side, pushes out the leaving group in one step. These three groups are pushed away from that, they invert, and we get this product. I'm going to discuss with you two important reactions of nitriles. The first is the acid-catalyzed hydrolysis of nitriles. We actually discussed this earlier in this section, earlier in this chapter. We saw that we could take a nitrile, we could put it in with aqueous acid and heat, and we would make the carboxylic acid. This is, again, a classic PRAM mechanism heteroatom exchange. The phases were add water, and then remove NH2, and in fact, the mechanism is fully covered in an earlier section of these notes. We can also do hydride reduction of nitriles. In order to do this, we do have to use lithium aluminum hydride. And what we observe is when we use lithium aluminum hydride, we get a hydride adding once, adding twice, and then when we put in something to protonate, to hydrolyze, we get two hydrides on the carbon and two hydrides attached to the nitrogen. So this makes a primary amine like that. Again, we use water here when we make amine products because if we used hydronium, the amine product would do acid-base reaction with the hydronium, and we would get the ammonium, which is not the end of the world, but it's likely that we would want the amine. Finally then, I briefly want to discuss with you a very special carboxylic acid called carbonic acid and talk to you about some of its important chemistry, particularly in biochemistry and industrial chemistry. Now, I first want to discuss with you the name carbonic acid. I want to just go back and review a little bit about how chemists initially named the inorganic acids. So for example, if we look at this structure, this is the octet rule violating structure of sulfuric acid. We can see we have sulfur, we have a double bond to oxygen, and then we have OHs directly attached. So sulfur in the middle, double bond O's, and OHs. This was called sulfuric acid, right? Because we have a sulfur in the middle and then it's an ic acid. Similarly, we did briefly reference the chromium equivalent of this. We had chromium in the middle. We have double bond O, OH, and OH. We called this chromic acid for the chromium. So when scientists began to look at the equivalent structures for carbon, they found a potential structure like this. C double bond O, see carbon in the middle, double bond O, OH, OH. And so they called this carbonic acid. Carbonic acid. So you see this name is basically sort of equivalent to the way we would name an acid that had sulfur in the middle or an acid that had chromium in the middle. We we'd have carbonic acid, and in fact, there are a number of other acids that are named this way. Now, carbonic acid is very interesting because it's actually unstable in acid. Now remember, carbonic acid is itself an acid. So when we put carbonic acid in acid, in other words, when we make more than one molecule of carbonic acid, that acid will catalyze the decomposition of carboxylic acid and the formation of carbon dioxide and water. And essentially what happens here is one of the OHs gets protonated, makes it a water-leaving group, the water-leaving group leaves, we get a protonated form of carbon dioxide, we deprotonate, there's our CO2. So that means that we can actually never have carbonic acid itself in a bottle. It's just going to decompose rapidly. But we can have carbonic acid as an intermediate if we put it into into strong base. So this leads to this super interesting biological application of carbonic acid. Carbonic acid and CO2 are used together as a buffer to maintain and regulate the pH of all of the aqueous materials. So that's the blood plasma, the cytosol of the cell, the interstitial fluid. All of these aqueous environments have a pH that's maintained by the presence of carbonates and carbon dioxide. Now here's how this works. We essentially have an equilibrium. We start with looking at the equilibrium of carbon dioxide plus water. So if we mix carbon dioxide and water, a small amount of that is going to react and form carbonic acid, which we know is unstable and is going to decompose readily back. So we would get this rapid equilibrium formed. However, if there's some base present, the base can react with the small amount of carbonic acid and form this. This is hydrogen carbonate ion. Hydrogen carbonate ion is stable. So, in a situation where the pH becomes too high, there's too much base in that aqueous solution, because there is carbon dioxide naturally present from our metabolic processes, some of this small amount of carbonic acid will do acid-base reaction with that base to bring down the pH to its desired level. And you would end up with some relative concentration here, which would essentially, using Henderson-Hasselbalch equation, we could calculate exactly what its pH would be, and we would know what our concentration was. Now, once we've established this buffer then, in the case where we started to release acid into that system, our pH became too low. And so we see this, for example, if we go into starvation, metabolism, or things like that. What we would see is that the excess acid would do acid-base reaction with this hydrogen carbonate. It would make carbonic acid, and then that would decompose and release more CO2. So therefore, this system right here is a very effective buffering system to regulate the pH in larger organisms like mammals. Interestingly, many larger organisms also use this CO2 for other purposes. So one of the issues that larger organisms have is that when they metabolize things like proteins, they begin to create ammonia. Ammonia is actually somewhat of a problem. It becomes toxic if too much of it is around. It affects the pH, it's a nucleophile, has a lot of bad effects. Now, the way that most biological organisms, larger organisms, dispose of waste products like ammonia is that they would put them into the bloodstream. The bloodstream would move to some type of organ which would filter out the undesired waste products. So for example, our kidneys are used to filter out waste products, okay? And then they would be discarded either in the urine or in other ways like that. The problem is that the biological organism is not going to want to dump large quantities of ammonia into the bloodstream because ammonia is toxic. So all it would be doing is shipping that ammonia around to all of the rest of the organs and cells in the body and possibly killing those cells and killing itself. So what it wants to do is it wants to essentially create a chaperone molecule to absorb the ammonia, create a molecule which is not as toxic as ammonia, that can then be water soluble, shipped to the kidneys, and be removed by filtration. The molecule that has evolved for that process in mammals is urea. And if we look at urea, urea is actually a double amide of carbonic acid. We take carbonic acid, we replace each of the OHs with an NH2 derived from ammonia. So one carbonic acid can remove 2 moles of ammonia. It becomes urea, and it forms just water as a byproduct, so that's very non-toxic. And then the urea is put into the bloodstream, it's shipped to the kidneys, it comes out in our urine. So this is a really interesting example of how natural systems have been optimized through evolution to be efficient rather than creating an entirely new molecule to aid in the disposal of this waste product. It uses carbonic acid, which is itself created from a waste product of metabolism. It's very efficient. I should also point out that this works very well in organisms that have access to large amounts of water, but there are organisms, birds, snakes, that actually don't want to consume the large amount of water that's needed to ship the urea and filter it out. So in those organisms, a similar molecule is used called uric acid. It's basically a urea with a ring here. And uric acid is not highly water soluble, so it forms a solid, and so then the waste product is excreted as sort of a form of poop. So in fact, bird poop is actually largely bird pee, essentially, because it's largely composed of uric acid. Finally then, I just briefly want to discuss the esters of carbonic acid. So remember, carboxylic acids can form esters. If we take a carbonic acid and form an ester, because we have 2 OHs, we're going to absorb 2 molecules of alcohol, and we're going to make a molecule like this where we have C double bond O in the middle and 2 alkoxy groups. This is called a dialkyl carbonate. Remember, when we name esters, we use the carboxylate name, so we take ic acid off and we put 8 on there, that's carbonate, and then we name the carbon groups that are attached. So a carbonate is a diester of a carbonic acid. Carbonates are actually very important molecules in industry. Diethyl carbonate itself, it turns out to be a very useful molecule for adding carbons to other molecules, and it in fact appears both in synthesis situations and potentially in biological systems. And we can also make really interesting carbonate esters if we use diols and we attach one end of the diol to one carbonic acid, the other end of the diol to a different carbonic acid, then that leaves an OH which could attach to another diol and so forth, and we can make these long chains of carbonic acid carbonate ester, carbonic acid carbonate esters. These are called polycarbonates, and this is a type of of what we call a polymer, and it's used in plastic. In fact, the see-through plastic water bottles are actually made up of polycarbonate. So again, it's a very rich area. It's an area that we don't have a lot of time to discuss, but I just wanted you to be familiar with some of the basic terminology and some of the basic uses of carbonic acid. That concludes our series of lectures on chapters 20 and 21.